Can you clearly see my screen?
Yes, we can see it.
Yes, we see.
Thank you for your confirmation. Okay, good morning, everyone. Today I'm going to talk about the segmentation of total cell area in Brightfield microscopy images. In this study, we analyze methods based on each detection and morphological operation, which highlights areas of varying brightness across the image. Um, Uh, this leads to the creation of binary image, which separates total area occupied by cell from the background using Brightfield channel image. Now we're going to talk about the procedure, and we will describe the steps to perform these methods. The procedure was carried out with the feature software, although it can be applied in another image processing software if it includes functions such as edge detection and morphological operation. Let's see each step of the procedure. The first step, you need to open Brightfield image and equalize its histogram. Figure 1 provides information about Brightfield channel of HCT 116 cell line. You can see here in A an edited picture and in B picture with equalized histogram. And the second step is to perform canny age detection with Gaussian kernel radio pixel resolution set according to the table 1. This table shows recommended values for pixel resolution dependent filters. At the same time, figure 2 shows the age detection using canny algorithms. On figure 1, on figure 1b, using Gaussian kernel radius of 1.75, low threshold, varying values for high threshold, such as in A, high threshold set to 20, and in B set to 2, and in C set to 8. The source step is to run maximum filter with radius phi according to the last table, the table 1. In the H image, this filters fills most of the intercellular space as shown here in figure 3. In A we can see H image before filtration, and in B we can see image after filtration using radius 5 pixels. And in 4 and 5, which we should perform closing and opening morphological operation with the number of iteration and consent. This not only fill the remaining small holes in the image, but also eliminates debris localized outside the cells. And number 6, it's an optional step. It reduces the effects of morphological dilation introduced in the step 3 by maximum filter, as you see here in figure 4. In A, closing for filling holes, and in B, opening for removing debris, and in C, erosion to shrink the volume of segment area. To corroborate the precision of segmentation, overlay the outline of segmented areas with the original Brightfield image, as shown in figure 5, original Brightfield image outline with segmented area. Regarding expected results and discussion, figure 6 show the results of segmentation of 6 cell lines and with various morphologies. You can see here, for example, in B, MCF7, and in any NDA and NB 200.01. Since the procedure is based on age detection, it works based on cell type with clearly visible borders such as MCF7 and HT2029. On the other hand, figure 7 show picture of normal human dermal fibroblast. In A and B, all the all the remaining parameters were kept at default values for section 3. As can be seen in the picture, the processor is a little too rough to precisely copy to the shapes of the cells. It should be noticed that the procedure requires image acquired with a properly set up microscope and with decent contrast. Examples for poor quality image are shown in figure 8. In figure 8 and B, were acquired with insufficient illumination with B being out of focus, which lead to a strongly pronounced noise that is incorrectly recognized as a cellular structure. However, as can be seen in figure EF, image with less contrast, inequalities in illumination are processed with any false detection. To conclude, precision of segmentation was validated against manual segmentation performed by two biologists on the set of 10 images of various sizes and cell types. Disabled to provide information about the results represented as a mean of difference between automated and manual segmentation. The automated segmentation turned out to be more tolerant than manual, resulting in 7.9 and 8.5 positive difference. So that's all. Thank you for your attention.
Okay. 1 minute, please. Um, uh, my presentation is one chip analysis of C. elegans, muscular force, locomotion patterns in microstructured environments, understanding between an organism and its environment is imperative in biological process. In the study of the muscular forces of C. elegans and its locomotion, speed, uh, amplitude, and wavelength are observed. In the test, the maximum force exerted by the middle part of the worm with the chip was evaluated. The locomotion of C. elegans depends on the surrounding environment. C. elegans kept the sinusoidal motion on the device. Locomotion can be studied in various substrate environments. What else in this part in figure 1? These are cis-touch receptors throughout the body of C. elegans, responsible for the sensation. The touch receptions are located close to the cuticle of the body, running along the muscles of the body wall in the attempt to relate this muscle with the force generated. Um, what else? The positions of the pillar was compared with the receptor neurons. Um, in this part, figure 2, the device consists of 9 per 9 millimeter with a square matrix surrounded by pillars with PDMS. Elastat enclose the upper part. Each column functions as a beam that measures the force in each C. elegans. To see the movement, of the worm, a Nikon fluorescence microscope was used under 106 augment videos obtained at 5 hertz were processed offline using image processing. The chip was washed with water before each sample to avoid contamination. In this part, two different abutment configuration were used, hexagonal shaped pillars in A and C, and the pillars in the square structure in B and D. Okay, and this part in this part show the pattern of generating force by a single one with a column center to center distance of 140 millimeters, pillars 4 and 5 that were in the middle of the body producing the maximum force. This indicates that throughout the movement, any pillar that is in contact with the middle part of the worm's body registers the greatest force. And conclusions in this article, the locomotion force was demonstrated in C. elegans using PDMS pillars, and this means that the microstructured environment significantly affects the force of contraction of the worms. Thanks.
Okay. Good morning, everyone. Today I'm going to talk about this article called Skin on a Chip. Model simulated inflammation, edema, and drug-based treatment. So here we have, I want to start with this schematic. So the idea of this paper is to simulate the skin. So as you know, our skin it comprises three layers: the epidermis, dermis, and the endothelium. So the idea is that here they use three PDMS slabs, and then they are joined by these membranes. These are PET membranes, and they have a special procedure to bond them to the PDMS slabs. So here in these spaces, in these three layers that are formed, they simulate the epidermis, the dermis, and the endothelium. So here we have the structure of the device. As you can see in the section A of this figure, you can see that the layers, so let's look at B first. Here you see how it is assembled. So you know that here are the three PDMS slabs, and in between we have the PET membranes. So after it is assembled, they have a color here, use three dyes so that we can see how they are separated. And then if we look at the cross section, A prima to A, then you can see the cross section, and it is in the section D, you can see the the scanning electron micrograph image. So the three layers, and also in C, you can see the image of the membrane. Okay, so the objective is to culture three different cell lines. So first, to represent this part, the epidermis, they are going to culture human epidermal keratinocytes. So here is the A part. You can see how they grow, and also here in the red lines, you can see how in which layer they are. So they are in the first one. And then in the second layer, we have the fibroblasts, and it's represented here at the blue level. And then here in the third level, we have the Hubert cells, which represent the endothelium. Okay, and the last picture, you can see both cell lines. You can see how they grow, and they achieve the in 3 days they grew and formed a monolayer. Okay. Also, they stained these cells cells with different cell trackers so that we can differentiate between them. And you can see here a zeta stack of the fluorescence image so that the cells are attached, they are adhered to the PET membranes. And the idea is to simulate inflammation. So they study treatments for inflammation as dexamethasone, and they measure the RNA expression of inflammation, of these inflammation markers. So here you see when they have the control, the tumor necrosis factor alpha, which promotes inflammation, and then with the treatment with the dexamethasone. And they discovered that here the junctions between the cells, so here in the first row, you see the DAPI, the nuclei, and then in the second row, you see it here, they stained the proteins that allow the cells to be very close to each other, so the junctions, and these proteins are the succula occludens. And here you see that in the control, the junctions are very tight. But when you apply this tumor necrotic factor, then the junctions are disturbed, the structure is disturbed. That's why the inflammation is produced. And then when you use the dexamethasone to prevent as a pre-treatment, these junctions are protected. So here we see the merge of these two images, and that's how they may produce this model of the skin in the chip. And the idea is to replace the animal experiments, the experiments made in the animal model, because this chip is promising to, for example, for cosmetic industry and for analyze the different pharmaceutical products for inflammation. Okay, thank you very much.
Good morning, everybody. Today, I'm going to present this paper. In this case, I'm going to present a deep learning tool in order to predict the severity of Parkinson disease. In this paper, the author presents another application of machine learning, in this case using deep neural networks, in order to define the severity of this disease. Parkinson has different forms in order to detect this disease. For example, in the difficulty of speaking, writing, and walking. And the patients, in this case, present a problem in your neurons. So the deep learning, in this case, is used to evaluate the tool for predict the severity. One thing important in this paper is the voice in order to define the severity of this disease. For example, in this paper, the patient's voice is used to evaluate the effect of disease, because the motor sensitive in the patient can be difficult in the time according to the disease. For example, many researchers are developed in order to evaluate this tool. For example, different machine learning algorithms are used. For example, fuzzy means, supervector machines, neural networks, and other cases. A methodology of the author proposes in this research is the use in first stage the normalized data. For example, in this case, min-max normalization. the vectors of features is used according to the patient, used the voice, and the block diagram is described in this image. For example, the first stage is data collection. The preprocessing of data is it's an important role in this case because the media audios are reprocessing and filtering in order to reduce the noise. The next stage is the building the algorithms of, in this case, deep learning. The next stage is training the network, and finally is testing and create the evaluate of a final stage. The implementation is developed using TensorFlow. TensorFlow is a library designed by Google, and the code is written in Python scripts. The data collection are used in 42 patients, and the amount of recordings is half of 5,000 files. So in this equation, the normalized values are presented. In this case, the min-max rule is used to evaluate to improve the performance of data. The stage of deep learning is used to improve the accuracy of detection, and finally the improve to classification stage. 80% of data is used for training, and 20% is used for evaluate the architecture of this algorithm. In this table, the metric used to evaluate the severe and non-severe Parkinson disease in patients are present. The metric is evaluated above of 25 or above to 20. In the range, the non-severe is classified according to the value. In this image, the authors present the architecture of this algorithm. The input stage of this algorithm is designed with 16 neurons. This input layer is the features voice of the patients, and finally, the final stage is composed by two neurons, which allowed the classified information according to the severe or non-severe stage. Finally, in order to evaluate this proposed method, the ANFIs used supervector machines which are the normal method used in this case, are compared with a new method, and the accuracy in this case is above to the normal method. And finally, in order to evaluate the classification of algorithms, the method presented in this research also improve the results of a normal method. In conclusion, the deep neuronal network can be applied in order to classify different information. In this case, the audio files are classified to define the Parkinson disease with high accuracy. Thank you.
Okay, hi everyone. I'll be presenting this paper, Transforming a Well into a Chip, modular 3D printing microchip, microfluidic chips. So to capture the benefits of transwell inserts and organs on a chip, While overcoming their individual and shared limitations, they use 3D printing to develop an easy-to-use, customizable microfluidic chip that, similarly to Transwell, can be inserted into any standard culture platform. Each insert chip contains a cell culture chamber with an external diameter customizable with a capacity of up to 2 milliliters of cellular medium. Inlet and outlet channels on the upper part of the chip enable the chamber to be connected to the control flow systems, and the bottom part of the chip includes 4 small modular legs. The insert chip has several design aspects that overcome the current limitation of organs on a chip. The first one, the insert chip, is a standalone platform that can be integrated into almost any standard culture platform. This feature enables cells to be cultured without undergoing special optimization procedures. To achieve a straightforward integration into standard culture platforms, they designed the insert chip to be self-supported in 4 short legs. So this figure shows an example in which the length of the insert chip's legs is adjusted to change the distance between the membrane and the bottom plate. They designed the insert chip to have a porous membrane that allows the possibility to create gradient and diffusion between different cell cultures. Furthermore, it allows up to 3 different cell types to be cultured: on the top of the membrane, on the bottom of the membrane, and on the bottom of the well. The insert chip was designed in configuration that enables different flow configurations and shear forces to be induced in the cells. The insert chip was designed so that it can be fabricated by a regular 3D printer using transparent materials such as clear dental resin. Moreover, the insert chip is designed in such a way that the membrane can be easily disassembled, enabling cells to be imaged at high resolution. This feature enables the insert chip to be reused, making it cost-efficient. The use of 3D printing enables the design to be of the platform to be quickly modified, and it reduces the need for multi-step fabrication. When the insert chip is made of 3 parts: the base, the membrane, and a sealing ring. The base is fully made with a 3D printer. The membrane is interfaced into the insert chip with a ring made of PDMS. And to ensure complete adherence between the membrane and the sealing ring, they use plasma and aptes. So the chip is modular and can be integrated with existing platforms, and the versatility is especially important for controlling the diffusion, material gradient, and shear forces between the upper and the lower compartments. Moreover, the multiple insert chips are placed next to each other so that they can create multi-organ chip systems. So they also sought to demonstrate that the use of the insert chip as a modular epithelium on a chip and endothelium on a chip, and they monitor cell growth and barrier development over 4 and 9 days until the cell form complete confluent monolayers, and the barrier function was further tested via immunocytometry. To validate the platform, variability measurements were done without the cells and with the cells that were cultured with and without flow, and it can be seen that the cells created a barrier layer that flow induction enhanced the barrier properties. As I said before, they designed this insert chip in a way that the membrane can be easily removed from the chip after the culture period due to the presence of the PDMS ring by simply using a tweezer. Once the membrane is removed, it can be placed on a glass cover slip, and it can be perform any kind of immunochemistry. They also designed the fabricated the reducer made of PDMS that easily can be placed in the chip, reducing the active surface area up to 20%, and allowing channels to be created in any desired shape. And so different designs can be used inside these insert chips. And finally, they used this insert chip and integrated it into a commercial multi-electrode array platform. They use cubic cells inside this insert chip. When the cells created a confluent monolayer, the chip was placed on top of the commercial array. and cultured with hippocampal neurons. And as you can see in the picture, barrier permeability was monitored together with neural electrical activity. So it is important to note that these experiments are usually challenging to carry out in standard organs on a chip because not only because of the technical aspect, but also because of the biological aspect, which requires that both cell populations be at the same stages of maturation and functionality, which might be hard types of platforms. So this is the paper. Thank you very much.
Okay, thank you, everyone. Do you have any questions? Okay, I have a question for Ramiro. Ramiro, when they collected these voice recordings from the Parkinson patients, I couldn't understand well how they rate these. How do they obtain this measurement of how severe is the disease? Could you please elaborate a little bit about it?
Okay, in this paper, the 42 patients are selected from a specific hospital, and the audio files are recorded in different days with a specific task. For example, in this case, 45 activities are described in order to obtain 5,000 voice recordings. So the activities are described for a specific voice activities. So the sets in the processing are described in features, which include the filtering information and stack principal components of these files. I hope that I asked your question.
Yes, thank you very much. Thank you for your answer. Okay, does someone else have a question? All right, so if there are no more questions, I thank everyone and see you next week.
See you next week.
Okay, see you.
Thank you.
Bye-bye.